Welcome to another lecture by MedicoMedics. Cell Biology, Chapter 6, Cell Cycle and Division. In this lecture, we will discuss the importance of the cell cycle, stages of cell cycle. We will mention mitosis, the steps of cell division, regulation of the cell cycle, disruptions, and then compare mitosis with meiosis, and finally end with a summary. Now, what is the importance of the cell cycle? It's central in growth and development, so it enables organisms to grow from a single cell to trillions of cells. Its involvement in tissue repair and replacement, so it replaces old or damaged cells. Reproduction. For single-cell organisms, division is a way to reproduce. Furthermore, what is the clinical relevance? Well, dysregulated cell cycles can lead to diseases like cancer. Now let's explore the stages of the cell cycle in more detail. When we talk about the stages of the cell cycle, there are some new words that are all very high yield. As a student, you need to repeat these sections a few times to make sure that you remember these orders. So first we have the interphase, which is the preparation for division. And it consists of the G1 phase, or gap 1 phase. Here, cell grows and performs its normal functions. Then we have the S phase, or synthesis, where DNA is replicated. Then we arrive at the G2 phase, where cell prepares for division by making proteins and organelles. So looking at the cell cycle illustration here, we have the G1 phase, then we have the synthesis phase, we have the G2 phase where it's preparing for division, and then we arrive at the M phase, also known as the mitotic phase. This is where the cell divides into two identical daughter cells. So it includes mitosis, nuclear division, and cytokinesis, cytoplasmic division. So think of the interphase as the cell getting ready for a big event, and the M phase as the main event, where it splits into two. Now let's take a closer look at the events of mitosis, the most dramatic part of the cycle. So mitosis, steps of cell division. Now, just like we discussed the different steps in the interphase, in mitosis, we also have several phases. And again, these need to be repeated and studied carefully. First, we have the prophase, where chromosomes condense and become visible. A nuclear envelope begins to break down. Spindle fibers form. So looking at the illustration here, we have the prophase up here. Now we move to the second stage, which is the metaphase. And what is happening here? So the chromosomes attached to spindle fibers are lined up in the middle. So the chromosomes align in the middle of the cell. It's also called the metaphase plate. After this alignment is made, we then move into the anaphase, where sister chromatids are pulled apart to opposite poles by spindle fibers. So here, you see, chromosomes have become condensed and visible. The nuclear envelope has begun to break down. These spindle fibers are formed. These fibers then come into action. First, the chromosomes are aligned in the middle of the cell, and then they are pulled apart by these spindle fibers to the opposite pole or side. After that has happened, we then enter into the telophase. So the chromosomes now decondense. In the prophase, they condensed. Now they become decondensed. The nuclear envelope is reforming around each set of the new chromosomes. Why are there two sets? Well, we said that in the anaphase, they were split apart into into its own side or opposite poles. Now we're starting to see the image of two different cells appearing. So we have now the final part, which is indeed cytokinesis. And this is where the division of the cytoplasma occurs, making two cells. A mnemonic I myself used in med school. I just read it backwards and it kind of worded out stamp. And I thought, well, when we're talking about cell division and we're talking about diseases like cancer, etc., the word approval comes to mind. And you will learn in more detail going forward that during the cell cycle, there are checkpoints at place that allow for this one phase to enter into the next one. So it's constantly about approving something so it doesn't mutate into cells we don't want. And this is where you need a stamp of approval. And you just read it backwards. You get prophase, metaphase, anaphase, telophase, and cytokinesis. Maybe it's helpful to you too. And then we arrive indeed to the regulation of the cell cycle. So just like I mentioned a second ago, we have checkpoints in the cell cycle. So we have at G1, we have the G1 checkpoint, which ensures the cell is ready to replicate DNA. So we have here a checkpoint. After that, when it's approved, it goes into synthesis, right? After its synthesis, it arrives at the G2 checkpoint. And this is to ensure that the DNA that has been replicated was successful. And once that has happened, we then move into the mitotic phase, and we have the M checkpoint. And this ensures that chromosomes are properly aligned. Now, we can also mention regulatory proteins. So cyclins and cyclin-dependent kinases control the progression of the cell cycle. We also have tumor suppressor genes, which are there to halt the cycle if errors are detected. Now, what happens when these regulatory mechanisms, right? What if they fail? Let's explore a bit disruptions in the cell cycle. So what we could have is uncontrolled cell division. This would then result in cancer, caused by mutations in oncogenes or tumor suppressor genes. Another is cell cycle arrest. So the failure to progress through the cycle can lead to cell death, apoptosis. What is clinical implication here? Well, chemotherapy targets rapidly dividing cells by disrupting their cycle. Remember, if we have cancer, we know that there is an uncontrolled growth happening. We want to stop this. So chemotherapy targets these rapidly dividing cells and disrupting their cycle, leading to a halt in this division. And here we have an illustration, right? So normal division, one cell is divided into two. After in cytokinesis, this one cell then enters into the interface again, and then goes into the G1 phase, eventually going to the mitotic phase. And inside the mitotic phase, remember stamp, we read backwards. We go through the prophase, we go through the anaphase, etc. Leading to two new cells, and so on and so on. However, when we have cancer cell division, as you can see, it becomes this thing where uncontrolled cell growth stuck together eventually lead to the formation of a tumor. And if this thing then spreads and invades other structures, now we're talking about the cancer has metastasized. So it's invading other structures, it's spreading. Now, when we talk about mitosis, which is central for cell division, we have another one that we're going to now contrast mitosis with, which is meiosis. And meiosis is crucial when we're talking about reproduction. Now let's just briefly compare mitosis with meiosis again. So mitosis produces two identical daughter cells used for growth and repair. Remember, the key thing to remember is that mitosis produces two identical daughter cells. Meiosis produces four non-identical gametes, so sperm or egg. It also reduces chromosome number by half for sexual reproduction. Now you can look at this illustration here where it says mitosis 2n divides into 2 and then that divides into 2. But in meiosis, you see 2n here dividing into 1 and 1, etc. Now what does this 2n mean here? Well, 2n here represents the diploid chromosome number in a cell. Diploid means the cell contains two sets of chromosomes, one set inherited from the mother and one from the father. So in humans, 2n is equal to 46 chromosomes, 23 pairs of homologous chromosomes from each parent. Now, what does the N mean exactly? Well, the N represents the haploid chromosome number, meaning a single set of chromosomes, so no pairs. So in humans, N is 23. Or in 23 chromosomes. And haploid cells are found in gametes, so sperm and egg. And these are produced during meiosis. So the N here is 23 chromosomes. When we do the 2, we do simply, it means 2 multiplied by this, so 46. Now, that was about meiosis. Now, what about 2N in mitosis? So for a moment, imagine we're using the same illustration. So mitosis. So the parent cell, a diploid cell, 2N, right, undergoes mitosis to produce two daughter cells that are also diploid. So 2N, 2N. Compared to meiosis, where 2N became 1N and 1N, meaning 23 chromosomes and 23 chromosomes. Here we still have 46 and 46. Now the chromosome number therefore remains the same. So key points here is mitosis produces two genetically identical cells with the same chromosome number as the parent cell. This is the parent cell here. So because these are quite important concepts and very high yield during your medical exams and studies, it is worth repeating one more time that the key difference between meiosis and mitosis lies in their purpose and outcomes. So mitosis results in two genetically identical diploid daughter cells for growth, repair, and asexual reproduction. We're talking about this side. While meiosis produces four genetically unique haploid cells or gametes for sexual reproduction. Mitosis involves one round of division, maintaining its chromosome number, while meiosis involves two rounds of division. We have meiosis 1 and then we have meiosis 2, reducing chromosome number by half and introducing genetic diversity through processes like crossing over and independent assortment. So in summary then, the cell cycle ensures proper growth, repair, and reproduction of cells. Mitosis and cytokinesis divide a cell into two identical cells. Regulation of the cell cycle prevents errors and diseases like cancer. So understanding the cell cycle is crucial for medical advances, including cell or cancer treatment. Excellent. That's the end of chapter 6. Continue now to chapter 7.